Welcome to yet another objective or two. This one focusing on amino acids and proteins. So, in terms of the objectives, you're actually looking at this puppy. Draw a generalized amino acid and identify the amine acid, also called the carboxylic acid, and the R groups. I find this one is rather unique amongst your objectives because your verb, get away, is actually to draw. So of all of the fancy biochemistry molecules you've seen, this would be the only one that you are held responsible for drawing. So different ways of looking at this. I'm just going to show you, say, the first 10 amino acids. There's obviously a whole lot more amino acids than this, but if you just look at amino acids, you should notice something that each of them has in common. And by the way, no, you don't have to memorize them, and no, you don't have to be able to draw them all. All you are responsible for being able to draw is a generalized amino acid. So, take a minute, look at these amino acids, and tell me, what do you find that each of them has in common? Well, I think you're going to notice that each has this NH2 group. NH2, NH2, NH2. That's called an amine group. And of course, this representation doesn't really show it, but it is actually, of course, nitrogen bonded to two hydrogens. Of course, the nitrogen is then attached to the carbon. So every amino acid has this amine group. This is a fancy way of writing it. Then there's something else that each of these has in common, and just to emphasize it, I'm going to switch colors. Let's go to red. Each of the amino acids has an acid group. Isn't that interesting? Amine and acid. So the acid group is actually called a carboxylic acid functional group. C double bond O, OH, carboxylic acid. So just by looking at the amino acids, you can tell that every amino acid has the amine group, every amino acid has the acid group, and then what in the world do you suppose these ones that are highlighted as light blue? Those are going to be what we call the R groups, and they can be replaced by any one of these. Glycine is the simplest amino acid simply because hydrogen is the R group. This methyl group, CH3, it makes this amino acid alanine. So this is just kind of an inductive way of getting at this objective. This, on the other hand, is the actual generalized amino acid. So again, you're going to notice the amine group, NH2. You're going to notice the carboxylic acid group. And then you're going to notice both of those are attached to a central carbon. Carbon always has to have these shared pairs of electrons, which are denoted by the bond. So carbon to hydrogen, you always have a carbon to hydrogen. And then the only thing that's going to change from one amino acid to the other is this R group. Okay, the actual objective, draw an amino acid. I would encourage you at this point to actually grab a piece of paper and attempt to draw an amino acid. Feel free to stop the video. I'm going to draw one just because they don't have to be pretty. Unlike the ones you're going to see in your textbook, you, in drawing an amino acid, will always start with a central carbon. And as I said, carbon has to have 4 lines coming out of it all the time, 4 bonds. So carbon is attached to a hydrogen. Then carbon is attached to an amine group. So that's nitrogen attached to 2 hydrogens again. And again, these are, of course, chemical bonds. Then the carbon is attached to an acid group, which is simply C double bond O, and then OH. And then finally, the only thing that is going to change in the amino acid is what we call an R group. And you can either just put an R in there, or if you want to make the simplest amino acid, just put a hydrogen. And that will make it glycine. Of course, you could make the R group any other fancy functional group, like a methyl group, CH3, now it's glycine, or alanine, sorry. Again, it doesn't matter. You don't need to memorize the names of any of these amino acids. You just need to be able to draw them. Right. So, even though it's technically not part of this objective, if you are already familiar with what dehydration synthesis or condensation synthesis is, this is just a reminder that when you have these amino acids, the formation of a protein is going to be the removal of water from two amino acids to form an actual peptide bond. Peptide bonds are a carbon to nitrogen bond. You've got rid of OH off of one amino acid, you've got rid of H off the amine group off the second. That bond produces a dipeptide. Then there is another objective here which asks you to differentiate. Again, that means tell apart the primary, secondary, tertiary, and quaternary structure of proteins. So, if you have an amino acid, you basically have a single amino acid, one. If you have a dipeptide, you have two amino acids. If you have three amino acids all joined together by peptide bonds, you have a tripeptide. More than three, we might call a polypeptide. And then anything greater than 50 amino acids, we're going to call a protein. So again, this is just a progression from the number of amino acids in a sequence. And again, proteins will end up taking many shapes, but the actual straight chain sequence of amino acids, say alanine, leucine, threonine, etc., we call that the primary structure or primary sequence. Straight chain of amino acids. So again, you see where one amino acid is ended here, another amino acid is ended here, another amino acid is ended here, and on you go from one amino end to the carboxylic end on the other side of the peptide. So that's the primary structure. When the hydrogen bonding actually begins to occur between amino acids in that primary structure, you have a tendency for proteins to basically form an alpha helix or a plated sheet. So I'm going to go back there. We obviously didn't stay very long on that page. So this is called the secondary structure. When you see an amino acid chain forming an alpha helix like this one, or if you see it forming a pleated sheet, we will call that the secondary structure. And that, of course, leaves two more possibilities. Amino acid chains can actually form two globular three-dimensional forms. One is called the tertiary structure. This would be showing, say, hemoglobin. Heme is a non-protein part of the hemoglobin molecule, but these protein chains are actually folded in on themselves to make a globular three-dimensional structure. So enzymes tend to have tertiary or what is called quaternary structure. Quaternary is very similar to tertiary. It's 3D, it's globular, but now you have more than one amino acid chain actually intertwined. And these become very important because it's actually the surfaces of these proteins that carry out catalysis or enzyme catalyzed reactions. So in the case of hemoglobin, you actually have 4 different protein chains that are all intertwined and functioning as one. So just to summarize, primary structure, sequence of a chain of amino acids. The secondary, you'll see the alpha helix or the pleated sheet. The tertiary, when basically the alpha helixes fold on top of one another. And then finally quaternary, more than one amino acid chain.